Among the many hidden roles of vitamin B12, perhaps the most underappreciated is its deep connection to DNA integrity and the cellular mechanisms that guard against aging. While B12 is often simplified as a red blood cell vitamin, its involvement in DNA synthesis and repair weaves it directly into the cellular machinery responsible for maintaining genetic stability. This subtle but crucial role gives vitamin B12 an unexpected place in the broader narrative of anti-aging science, not as a trendy antioxidant or a fashionable peptide, but as a quiet molecular custodian of longevity. When B12 levels remain chronically low, this oversight can lead to invisible genomic erosion and an acceleration of biological aging that unfolds slowly, yet irreversibly. At the biochemical level, B12 is essential for the function of methionine synthase, an enzyme that converts homocysteine into methionine. This reaction is not just about detoxifying homocysteine, a known neurotoxin, but about producing acetylmethionine, SAMe, the body's primary methyl donor. SAMe is required for methylation reactions that affect gene expression, epigenetic signaling, and DNA repair. Without sufficient B12, the methylation cycle slows, homocysteine accumulates, and the production of SAMe drops. This trifecta undermines the body's capacity to silence damaged genes, repair broken DNA strands, and maintain epigenetic programming that protects cells from senescence and dysfunction. What makes this particularly insidious is how these changes can occur without any immediate clinical signs. Long before anemia or neuropathy develop, B12 deficiency may already be disrupting the cellular instructions that regulate replication, apoptosis, and immune surveillance. DNA repair enzymes rely on methylation-dependent pathways to correct single-strand breaks and suppress genomic instability. Without B12, these repair systems operate at reduced capacity, allowing mutations to accumulate and damaged DNA to persist longer than it should. Over time, this results in a higher burden of somatic mutations, chromosomal abnormalities, and telomere attrition, hallmarks of biological aging that contribute to cancer risk, cognitive decline, and immune dysfunction. One of the clearest demonstrations of this process is seen in studies linking low B12 status to increased micronuclei formation, a marker of chromosomal damage, in blood cells. These micronuclei are remnants of missegregated or broken DNA that reflect faulty mitosis and poor repair. They're more common in older adults, individuals with poor methylation, and those with chronic inflammation, all conditions where B12 demand may be higher or absorption compromised. Interestingly, B12 repletion has been shown to reduce micronuclei counts, suggesting that supplementation doesn't just stop further damage. It may also actively promote repair and genome stabilization, even in aging tissues. There is also a growing interest in how B12 status influences epigenetic aging clocks, which use DNA methylation patterns to estimate biological age. Because methylation is central to both gene silencing and the epigenetic memory of a cell, insufficient B12 could lead to premature deregulation of gene expression, especially in genes involved in inflammation, oxidative stress response, and cellular proliferation. This implies that chronic low B12 might silently accelerate aging at the molecular level, even in the absence of visible symptoms. Conversely, optimal B12 status could support the maintenance of a youthful epigenetic profile, particularly when combined with other methylation cofactors like folate, choline, and betaine. This hidden anti-aging potential of B12 is especially relevant in the context of modern life, where many people, particularly the elderly, vegetarians, and those with gastrointestinal disorders, are functionally deficient without knowing it. Stress, alcohol consumption, certain medications, and genetic polymorphisms affecting methylation pathways, such as MTHFR mutations, can all increase the body's need for B12. Yet routine testing often focuses only on serum levels, which may not reflect intracellular availability or functional capacity. It's entirely possible for someone to carry a normal B12 level on paper while silently accumulating DNA damage, methylation errors, and epigenetic drift that accelerates aging from within. From a therapeutic standpoint, this opens a compelling case for using B12 not only to treat deficiency, but as part of a broader strategy to preserve genomic integrity and cellular resilience. High-dose methylcobalamin supplementation, in particular, may support DNA repair mechanisms in neurons and other post-mitotic cells, which cannot be replaced once lost. When combined with a nutrient-rich diet, antioxidant support, and lifestyle factors like exercise and sleep, B12 may contribute meaningfully to slowing cellular aging and extending health span. Its water solubility and low toxicity make it especially attractive for long-term use, even in high doses. In the grand scheme of anti-aging interventions, B12 is not flashy. It does not promise miraculous reversals or dramatic transformations, but it operates at a level of fundamental biology that is non-negotiable. Without adequate B12, the genome becomes unstable, repair processes stall, and the epigenetic architecture of youth begins to decay. In this light, B12 is not merely a maintenance vitamin. It is a molecular safeguard, a quiet defender of our DNA's structural and informational integrity. To ignore it in the context of aging is to miss one of the most essential links between nutrition, genome fidelity, and the long arc of cellular life.